Hello, so we will be discussing today PYQs from plant physiology. So the first question, which of the following is not observed during apoplastic pathway? So the first one is apoplast is continuous and does not provide any barrier to water movement. Yes, it is correct. Movement of water occurs through intercellular spaces and wall of the cells. Yes, it is correct. The movement does not involve crossing of cell membrane. Movement is aided by cytoplasmic streaming. No, this is seen in symplastic pathway. Let's see the explanation from NCERT. So they have mentioned about the apoplast pathway, that this in apoplast pathway the system has adjacent cell walls that is continuous throughout the plant, as you can see in this diagram, except at the Casparian strips. So it is almost continuous through the intercellular spaces traveling to the cell walls. Here when it reaches the endodermis, it comes across there is Casparian strip which it cannot pass, then it again follows a symplastic pathway, but till there it is passing continuously through the intercellular spaces. Then the next point there also is mentioned over here, that it excludes that the movement of water occurs exclusively through the intercellular spaces and walls of the cells, as we can see in the diagram. Then the next point is also saying that there is no crossing of the cell membrane that is taking place over here. Then the symplast, explanation for the symplast. So the symplast system has cytoplasmic strands that extend through the plasmodesmata. So the movement of the water takes place from one cell to the next cell with the help of the plasmodesmata. So there is plasmodesmata present between the cell walls. Then during this symplastic movement, the water travels through the cells. They are cytoplasm, that is mean they're traveling from one cell to the other, and the symplastic movement is therefore aided by the cytoplasmic streaming. So we can say this. So this statement has been given about the symplast pathway. So here this pathway is the symplast pathway. This is the plasmodesmata present between the cells. Here this is the diagram. So here you can see through the intercellular spaces where the water is traveling through the walls. This is the apoplastic pathway. It is reaching here till the endodermis where there is Casparian strip, and till there it is continuous without any barrier. Then here in the symplast, it is traveling from one cell to the other, from one cytoplasm to the other through cytoplasmic streaming, because there is plasmodesmata that is present. So therefore the correct answer is the fourth one. Then the next question, production of cucumber has increased manifold in recent years. Application of which of the phytohormone has resulted in the increased yield, as the hormone is also known to produce female flowers. So they are saying, they're asking about a phytohormone which shows these physiological functions like production of the female flowers. It is also increasing the yield, and specifically they're talking about the cucumber with this example. So let us see about these, which one of them shows these functions. So here we have ethylene. So ethylene is responsible for initiating flowering. It is promoting female flowers in cucumbers and also increasing the yield. So this exact function has been given for ethylene. So the correct answer would be ethylene. Then next, which of the following produces nitrogen fixing nodules on the roots of Alnus? So they're asking about they have given us about the microbes. All these are microbes, and they have given us about this plant, that nitrogen fixing nodules is formed on the roots of Alnus. So which of the microbes is the one which is producing this nitrogen fixing nodules? Let's look at what is this nitrogen fixing nodules. So in this biological nitrogen fixation, it is a symbiotic relationship between the microbe as well as the root of the plant. So the most common example is the legume and the bacteria. So the legume plant and the bacteria. So in this case, we have the rod-shaped rhizobium, which is having a relationship, symbiotic relationship in the roots of legumes. Specifically, the examples like alfalfa, sweet clover, sweet pea, all these are given. But if we talk about another example, okay, this association is seen in the form of nodules. This association that is there between the microbe and the root, it is seen in the form of nodules, which are nothing but a small outgrowths on the roots of the plant. So another example is Frankia, which is producing these nitrogen fixing nodules on the roots of a non-leguminous plant. Example they have given is ulmus. So they have given the example of ulmus, and the microbe which is producing the nitrogen fixing nodules is the Frankia. Let's see more explanation. This is how a root nodule actually looks like. Then this is the diagram from NCERT. So here are the bacteria around the root hair. They're going to form an infection thread and they're going to reach the inner cortex, and by continuous cell division they will form a mature root nodule. So the correct answer is Frankia. Next is this question. Girdling experiment was performed by plant physiologists to identify the plant tissue through which of the following. So they're asking, this girdling experiment was performed to identify which of the functions of this plant tissue. So after this experiment, it was found that food is transported through phloem, to identify the plant tissue through which food is transported. So they were able to find out the tissue through which a food is transported. So this is how a girdling experiment is done. You can see this ring-shaped structure where the bark has been removed, including the phloem part. So from outside, if I start scraping this and I reach till the part where there is phloem, that also I will be removing the bark part, including the phloem part, and this much part will be remaining, and it will form a ring-like structure in the form of a ring that is supposed to be scraped out. So after a few weeks, this will be seen. This kind of a swollen structure will be seen because the food that was made was not able to travel in the downward direction towards the root. And since the tissue that has been removed is phloem, so it's understood that phloem is responsible for transporting the food in one direction, that is in the downward direction. Let us look at the explanation given in NCERT. So a simple experiment, that is girdling, was used to identify tissues through which food is transported. So on the trunk of a tree, a ring of a bark up to the depth till we reach the phloem layer, we'll keep scraping it out, is removed. Then in the absence of this downward movement of food, the portion of the bark above the region on the ring will be swollen after a few weeks, because now the food has been accumulated over here. So through this it is understood that the phloem is the tissue that is responsible for the translocation of the food, and the transport is taking place in one direction, that is towards the roots. Then the next question, what is the role of large bundle sheath cells found around the vascular? What is the role of large bundle sheath cells found around the vascular bundles in the C4 plants? So the first point that I have given is to protect the vascular tissue from high light intensity. This doesn't sound to be a proper valid reason for the role of the large bundle sheath cells. So let's see the other statements that are there. To provide site for photorespiratory pathway. The reason for having C4 plants, they are having a different anatomy as compared to C3 plants, is because they want to avoid photorespiration. So there is no point of performing photorespiration. So this point is absolutely wrong. To increase the number of chloroplasts for the operation of Kelvin cycle. Yes, this could be one of the reasons, because we want to carry out as much as possible Kelvin cycle and avoid photorespiration. So if there are large bundle sheath cells and they are showing increased number of chloroplasts, so they will obviously help in the operating the Kelvin cycle to avoid photorespiration. So yes, this could be the reason. To enable the plant to tolerate high temperature. Again, this doesn't sound to be a valid reason for those large bundle sheath cells. So this would be the correct answer. Let's look at the explanation. So in NCERT, they have given these statements stating that there are large cells around the vascular bundles in the C4 plants, which are the bundle sheath cells. They have the crans anatomy, and these bundle sheath cells form several layers around the vascular bundles. They are characterized by having large number of chloroplasts around them. They're having large number of chloroplasts inside these bundle sheath cells. So we can say that this could be the correct answer. Then this is more explanation. So anatomy of the leaves of C4 plants is different from the C3 plants, which is the Kranz anatomy. There are bundle sheath cells around the vascular tissues. These chloroplasts are large, while in mesophyll cells the chloroplasts are small. So we can see in the diagram over here, we have the vascular bundles. Around the vascular bundles there are these bundle sheath cells, and then after that we have the mesophyll cells around them. So now these chloroplasts of these bundle sheath cells contain what? Contain the enzymes required for the Calvin cycle, because of the high concentration of CO2, then the RUBP carboxylase will be able to participate in Calvin cycle, which is its main function, and then photorespiration can be avoided through this. So therefore the correct answer would be the third one, to increase the number of chloroplasts for operation of Calvin cycle. Now, addition of more solutes in a solution, given solution will not affect the water potential. No, it will affect the water potential. It will raise its water potential. No, it will lower the water potential. Yes, it will lower the water potential. It will make its water potential 0. No, first of all, it's already a pure water. So pure water is having only water molecules. It is free. It is having its maximum water potential. They are free. Water molecules are free, and the maximum water potential for a pure solvent is 0. If a solute is added, if I'm adding solutes, the water molecules will now be associated with the solute molecules. So the number of free water molecules will be lowered, and since the free water molecules will be lowered, the water potential will also decrease. If the water potential is decreasing, it will move towards the negative side. So in case of adding more solutes, the water potential decreases. So let's see, all these statements are I've taken from the NCERT. So the greater the concentration of water in a system, greater is its kinetic energy, which is nothing but its water potential. So a pure water will have the greatest water potential. And what is this water potential? In a pure state, it is nothing but, which is not under any pressure, it is nothing but, it is 0. The water potential, the maximum water potential. And if more solute is dissolved in this pure water, the solution has fewer free water molecules, and the solution has fewer free water molecules, and the concentration of free energy of water molecule decreases, thus reducing its water potentials. The water potential is decreasing, therefore the correct answer would be the third one. Then next question. The primary CO2 acceptor in C4 plants is phosphoenol pyruvate, that is PEP, and is found in the mesophyll cells. Yes, correct. Mesophyll cells lack Rubisco enzyme. So which one of the following applies for this? So statement 1 and 2 both are correct. Let's look at the explanation for this. So the primary CO2 acceptor is a 3-carbon molecule, which is phosphoenol pyruvate, which is nothing but PEP. So this we are talking about the C4 plants. The C4 plants show this Hatch and Slack pathway, which is there in front of you. This diagram is of Hatch and Slack pathway, which takes place in the C4 plants. Then the next point is saying that the mesophyll cells lack the Rubisco enzyme. So atmospheric CO2 which was present in the atmosphere, it entered first the mesophyll cells, which is there present over here. This mesophyll cells then got combined with the phosphoenol pyruvate, that is the PEP. It got fixed. The CO2 first, the fixation takes place of the CO2, which gives us C4 acid. This C4 acid in this then transported to these bundle sheath cells, where here now by decarboxylation the CO2 will be utilized for Calvin cycle. So here the Calvin cycle is going to take place, and here the C3 cycle is going to take place in the bundle sheath cells. So these statements have been given for Hatchen's-like pathway in NCERT. So this is the explanation. So the correct answer is that both these statements are correct. Then the gaseous plant growth regulator is used in plants. So they are talking about, they are not giving the name of the plant growth hormone, but they are giving us a hint that it is a gaseous plant hormone. So you should know that which is a gaseous plant hormone, as well as now according to this you have to match the functions, that it is which of the first of all knowing that which is a plant hormone which is in a gaseous plant hormone, and then when you are able to identify that hormone, then we can easily understand that from all these following functions which of the function applies for this gaseous plant growth hormone. So let's read. First of all, this gaseous plant hormone is nothing but ethylene. Now, to kill cotyledonous weeds in fields, this is performed by auxins, synthetic auxins, that is 2,4-D. Speeding up melting process is performed by gibberellins. Promoting root growth and root hair formation to increase absorption surface. This is exact line for ethylene which they have given the function of ethylene. So all these physiological functions of phytohormone have been given. And then help in overcoming apical dominance. This is a function of cytokinin. Let's look at the exact functions from where they have been picked up, from which line, from NCERT. So your auxin. So in auxin, indole acetic acid and indole butyric acid are the ones that have been isolated from plants, but in case of naphthalene acetic acid, NaA, and 2,4-D dichlorophenoxy acetic acid are synthetic auxins, and they are used for agricultural and horticultural purposes. What are these purposes? Let's see. They are used like in the form of herbicides. So how? 2,4-D is widely used to kill dicotyledonous weeds. Then in case of cytokinins, the exact line, these functions are given, that cytokinins help in overcoming the apical dominance. So this point is for cytokinin. Then for gibberellins, 1, 2, 3 are known. So gibberellin 3 is used to speed up the malting process in the brewing industry. In the brewing industry, malting process is done in which gibberellin is being used. So this point is for gibberellins. Then coming to ethylene. Ethylene is a simple gaseous plant growth regulator, and it is promoting root growth and root hair formation. Thus it is helping the plants to increase their absorption surface. So therefore this function which was given applies for, if you know these 2 points of gaseous plant growth regulator and the absorption by the root hair. So the third point would be, the third one is the correct answer. Then these are the plant growth mineral nutrients for plants, and these are its functions. So let's match these functions, these mineral nutrients, with their functions. So for boron, we know it is for pollen germination. Iron is known to activate the enzyme catalase. So if you see, C1 is present over here, C2 is present, and D1 is present in this one. So the third one should be the correct answer. Let's look at the explanations. What are the functions given in NCERT? So all these have been mentioned. In case of magnesium, it's mentioned that it activates the enzymes of respiration, photosynthesis, and is involved in synthesis of DNA and RNA. In manganese, it is given that it is involved in splitting of water to liberate oxygen. They have also given that it is also involved in activating enzymes involved in photosynthesis and respiration. So both of them have these functions. But here splitting of water has been mentioned for manganese. So this we will be matching with the splitting of water. And then the remaining, the enzymes of respiration and photosynthesis, will be given to magnesium. So now then the boron helps in pollen germination. Iron, that is also mentioned, it is activating in the catalase, and it is helping in activating the catalase enzyme. So let's see, look at the options. So A4, manganese helping in activating, A4, manganese functioning in the splitting of water during photosynthesis? Yes, manganese is helping in splitting of the water to liberate oxygen during photosynthesis. So yes, so for here the answer would be the third one. Thank you so much, and please do subscribe and like the video. If you like the video, then do please hit the like button.